We're doing highlights from chapters 3 and 4 in lecture today, and we'll be talking about how proteins work, specifically how enzymes work, and their similarities. We'll be talking and doing some math problems with enzyme kinetics and explaining how to quantify different parameters of enzyme activity and how neat that is. And then we will talk in sort of a global sense about how we control proteins and enzymes. Most of the enzymes that we're going to see this semester are proteins. So it'll be good fun romp. Here we go. So enzymes being a one of the functions that proteins have. Some proteins are enzymes, which means they have catalytic capacity. They are going to take a substrate and facilitate its conversion to product. And they do that by binding to the molecule, the starting molecule or the substrate molecule. And they, because that binding is very specific, like a lock and key, then for every reaction that you have where you have a substrate go into a product, every different reaction, every different kind of reaction is going to require a specific enzyme to catalyze it because of that binding specificity. So if you think about, in general, just proteins, any kind of protein that's going to bind with some other molecule, we'll call, in here we have the protein in green, and we have the molecule that we're going to bind that protein to in yellow. And so our generic term when a protein binds another molecule, that other molecule is called a ligand. And so when you bind the ligand, the shape matters because it's like a lock and a key. So the shape of the ligand is going to govern the kind of shape that you're going to have in your binding domain in your protein. And so for different kinds of molecules, you're going to have different proteins for binding because of that lock and key kind of interaction. And so what we see with proteins is that the binding domain is going to be lined with a particular series of amino acids that are going to facilitate the binding of that ligand once it's close. Typically these are non-covalent bonds, sometimes van der Waals forces, and sometimes hydrophobic interactions are going to help facilitate binding. So if we think of a protein, and we know that when we unwind its polypeptide structure down to its primary structure, we just have a linear chain of amino acids. And here we've picked out and highlighted specific amino acids along this chain. Most of them we haven't shown here, but for the ones that we have picked out and given a color to represent their R group, we see that when this protein folds into its secondary and tertiary structure, now these distant amino acids on the primary structure are now producing the binding domain for the protein. And so that can become quite important if you think about this structure here being representing this protein. And then here is our ligand that's going to bind, in this case cyclic AMP. Here is the ribose sugar and the adenine head group and the phosphate group, and the cyclic part folds back onto the sugar, and so it makes the cyclic AMP. This is a cyclic AMP molecule, and it's sitting in the binding domain. And if you remember the colored balls here, green, yellow, red, green, blue, green, and then you see they've colored the side chains here, green, yellow, green, green, yellow, red, green, blue, green, here they are. And so they are lining this binding domain in such a way that we get hydrogen bonding with our ligand, in this case the cyclic AMP. So the specific structure of the cyclic AMP is going to dictate how we need to produce the binding domain on the protein so that we can get these, in this case, hydrogen bond. Most of these are hydrogen bonds, but we actually have an ionic bond here between two charged portions of the atoms. And so this is going to have a nice stable interaction with the protein because of these hydrogen bonds and the ionic bond. So this is how binding works with proteins and their ligands. So because the enzymes that we are going to be talking about most of the time this semester are proteins. It's the same kind of interaction when you have an enzyme binding to a substrate molecule, and then that enzyme helps facilitate or speed up the reaction, and you get your product. And so it's about binding. So the enzyme is going to have a specific binding domain that we call the active site, where the substrate is going to attach to the protein. And so there are a variety of ways that we can have this lock and key interaction. Sometimes it's charge mediated, and when you get the opposite charges together, you can favor a reaction. Sometimes when you're trying to put two molecules together, the enzyme's utility is that it brings them in the same vicinity at the same time, and so facilitates their binding in that way. And sometimes when the enzyme binds to the substrate, it perturbs bonds in the molecule such that it favors a reaction. There are a number of ways that you can get these reactions to happen, but what's similar is that there's always a binding interaction between the substrate or substrates and the enzyme. And so when we have a binding domain in an enzyme that's specifically binding a substrate, we call it an active site. The active site is where the substrate or substrates bind to the enzyme. So here's another cartoon drawing. Our enzyme shown in green. Here's our substrate molecule shown as the red square, diamond, whatever that is. And so it binds in the active site of the enzyme. You have a temporary enzyme substrate complex. Then the catalytic reaction happens and we produce our product. There's a temporary binding of... Alright, so because the enzyme isn't altered in the process, it binds to the substrate, converts it to product, lets go of the product. We can use this enzyme many times before it's spent. And so while theoretically the reverse reaction works too, the enzyme binds to the product and convert it back into substrate. In reality, in cells in vivo, we set up the situation so that there's always a favoring the unidirectional process of a reaction, where we have the equilibrium set up so that we get the reaction happening in the direction that we need. And so a lot of times that means as soon as we create a product, then we convert it into something else so that we favor the unidirectional motion of this reaction, as opposed, even though in theory the same enzyme can also catalyze the reverse reaction. But don't get too wound up about that. All right, so let's have some fun with protein kinetics with enzyme kinetics. So if we think about a set given amount of enzyme, if we think about supplying that enzyme with some substrate, we know that we're going to get a reaction. And let's say that we have some sort of way to monitor the reaction. Maybe we get a color change when the substrate's one color and then it changes to a product. Maybe it changes color, so we can watch that progress and we can time how fast the reaction happens. So for any reaction, you can calculate the velocity of the reaction, or the rate of the reaction. And what you see for any given quantity of enzyme, as you add more substrate, you increase the pace of the reaction. As more substrate is available, here's our substrate, so we increase the amount of substrate available for a given quantity of enzyme, we get increased velocity of the reaction up to a point. So we can get more product converted, which is our measure of our rate of reaction, the more substrate that we have. Now, it gets to a point where it doesn't matter how much more substrate we add, we don't get a change in the velocity of the reaction. And that's because if you're talking about a set quantity of enzyme, then once you've added so much substrate that all of the enzymes are occupied, even if you add more substrate, if all your enzymes are occupied, then you've saturated the system, and you're going to reach a point where you get a maximum velocity. So that's called Vmax, the maximum velocity. And so you can do these experiments to demonstrate that as you increase the substrate concentration, you initially get a linear rate of increase in the velocity, but that eventually it levels off and plateaus, And when you've saturated all of your enzymes, then that's our maximum velocity. We can also identify another parameter called Km. Km is a constant. It's called the Michaelis constant. And this is a substrate concentration corresponding to 1/2 the maximum velocity. So if you've got a maximum velocity that's, say, 4, then at 2, at the velocity that's corresponding to the numerical half of that maximum velocity, whatever substrate concentration that corresponds to, we call that our Michaelis-Menten constant. So this is just a way to explain or describe how an enzyme works. We know our Vmax, and we can determine our Michaelis-Menten constant once we've done this series of reactions where we vary the substrate concentration. And so for any one enzyme, that may not tell you very much. You can figure out the Vmax and the Km, but where this really comes into use is when you're comparing different enzymes and different reactions and how fast they go and what their Kms are. And so sometimes you will see that there's a different maximum velocity for a quantity of enzyme and or a different Km, a substrate concentration corresponding to 1/2 Vmax for that reaction. And so then you can start to compare the kinetic parameters for different reactions. So if you have a low Km, that typically means that you have a tight binding of the substrate to the enzyme. It means that the enzyme has a harder time letting go of it so that it can't, it takes a while for it to get back and to churn out more, bind to more substrate. If you have a high Km, that means that there's typically a weak binding of the enzyme to the substrate so that it can complete the reaction and be ready to go again, so you get more product out in those cases. Now you can also take a reaction and add an inhibitor and see how that affects the kinetic parameters. There are a lot of things you can do when you get into a little more biochemistry. But let's think about it this way. If that previous plot that we were just looking at where we had the concentration, the substrate concentration on the x-axis, we had the velocity on the y-axis, and as we increased our substrate concentration, we saw that we increased the velocity of the reaction. And so we get this, what's called a Michaelis-Menten plot that looks something like this. You have a linear portion at the beginning, but then it levels off and plateaus as you get saturation. And so you can eyeball and kind of give a guess of the Vmax, and then do the math to do 1/2 Vmax, and then figure out your Km by guessing from the data on the Michaelis-Menten plot. But it's also useful to do what's called a double reciprocal plot, and that's the Lineweaver-Burk. The Lineweaver-Burk takes the reciprocal of the substrate concentration, so we have 1 over the substrate concentration, takes the reciprocal of the velocity, so that you have 1 over the velocity. So if you look at the units, if you follow the units always help you make sure you're working the math the right way. So in this case, it tells us that our substrate concentration units were in micromolar, and that our velocity was in micromoles per minute. Velocity is distance over time, and so in this case, how many micromoles of product were generated per minute, or how many micromoles of substrate we processed per minute. And so then the double reciprocal then, if you've got 1 over substrate, then you end up with 1 over your micromolar, so micromolar to the -1. For velocity, our units were micromoles per minute, so when we take the inverse, we get minutes per micromole. And the other thing, when you do the double reciprocal plot, if you follow the substrate concentrations and follow the colors of the blue, you see that when you do the double reciprocal, now our increasing concentrations go down to the left. And we have a nice, pretty line, which means we can get the equation of the line, and that helps us then have a greater accuracy in our estimation of Km and Vmax. So the equation of the line here is 1 over V equals Km over Vmax times 1 over the substrate concentration plus 1 over Vmax. This should be familiar. So if we have 1 over V equals Km, the Michaelis constant, over Vmax times 1 over our substrate concentration plus 1 over Vmax, this should help you harken back to the equation of a line. Y equals mx plus b. All right. So these refer to the same thing. Our slope here is Km over Vmax. Our y-intercept b is 1 over Vmax. So if when you take the equation and figure out the numbers that go with it, if you get the example on the next slide is y equals 0.2 x plus 3. That means that you can set these terms as equivalent to one another. Now, of course, here that means 0.2 equals Km over Vmax. But you have two unknowns here, so we don't want to do this one first. This one, we just say, okay, 3 equals 1 over Vmax. So now we've just got one unknown, and this is a lot easier. So we flip them both. So Vmax equals 0.33. And let's say that our units are what they were here, which means that our velocity is going to be micromoles over minutes. You have to know your graph to figure out what your units are. And so just follow the units that you're given on the graph. So here's your Vmax, 0.33 micromoles per minute. So now we know what Vmax is. We can plug that back into here. So now 0.2 equals Km over, do the substitution, 0.33 micromoles per minute. Km equals 0.2 times 0.33. Let's get our trusty calculator out here. So 0.2 times 0.33, 0.07, and our substrate concentration was in micromole. Right. So that's pretty straightforward. Here's another example. This one says if the equation that you get is y equals 0.4x plus 4, then it asks for Vmax first, remembering that this is 1 over V, this is Km over Vmax times, and the x is 1 over the substrate concentration plus 1 over Vmax. So 1 over Vmax equals 4. Vmax equals 1 over 4, which equals 0.25. And let's say that this time we had Michaelis-Menten, and let's say that our substrate was in molar. And let's say that our velocity was in molar per second. Let's say it's a fast reaction. Okay? Of course, you can predict Vmax, which would be whatever numerical value is here. 1/2 Vmax over and straight down gives you your Km estimation there. That's the way you would predict Vmax and Km from a Michaelis-Menten plot, like so. But when you do the double reciprocal, you get your equation of your line here. And so that's what we're working on. So if we know Vmax, 0.25, and that's velocity, so this is molar per second, then we plug that in. If 0.4 is our slope, and that's Km over Vmax, oops, we substitute, and go ahead and substitute 0.25. That means Km, 0.4 times 0.25, 0.4 times 0.25, 0.1 Km, and so that's molar since our units in this example that I made up were molar. All right? So those are how we do calculations for the Lineweaver-Burke double reciprocal, and the estimations for Michaelis-Menten for Vmax and Km. Practice more of those problems. I'm sure in SI you'll be doing more practice also. So more information about how enzymes work. If you think about a reactant or substrate molecule Y that you need to turn into product X, there's a certain amount of energy that has to be input into the system in order to make that reaction go. And so we call that amount of energy is the difference between the substrate at its starting energy level and the energy that it has to have before it gets converted into a product. That's called the activation energy. It's the energy required to make Y go to X. And when you don't have an enzyme in an uncatalyzed pathway, that energy hill that you have here, this activation energy, typically means that you're going to have a temperature increase that would be above what would be tolerable or viable for the cell. So most reactions don't just happen spontaneously by themselves because they kill the cell. So what we do with the utility of enzymes is that by binding to the reactant, by binding to the substrate and facilitating the conversion to product in the various ways that they do it, it means that you have to put less energy into the system to get the reaction to go. So for catalyzed pathways, enzyme catalyzed pathways, there's not as much of a temperature increase that has to happen in order to make the reaction go. And so the utility of enzymes is that they lower the activation energy. So enzymes reduce the amount of energy that you have to put into a system to make it go, and that represents changes in energy in the cell that are viable and tolerable, usually. So in the case that they've shown here, we have an exergonic reaction. Our products have less energy than our reactants, and so in the process of doing this reaction, we've lost some energy here. The laws of thermodynamics tell us that we can't have completely efficient conversions of energy from one form to another. So we always lose a little bit of energy every time we convert molecules from one form to another. And so when we have a reaction that releases energy, it's called an exergonic reaction. Ergonic meaning energy, and X meaning out. So energy out is where we have products that have energy that's lower than the reactants. So some of that energy gets lost from the system to make it happen. Either way, the enzyme is still helping the reaction happen by lowering the activation energy required to make the reaction proceed. Now, endergonic reactions have the same requirement of enzymes to lower the activation energy. But the difference here with endergonic reactions is that our products have more energy than our reactants. So if we look at our energy hill diagram, where we had our total energy going this way, and we have our reaction pathway going this way, here we're going to have Y, or our substrate or reactant, whichever term you prefer, going to X, and X is going to have higher energy. So it's, I'm going to draw them on both on one set here. So no enzyme here. Our E sub A is quite large. We're going to have to put a lot of energy in to make this go. With a catalyzed reaction with the enzyme, we don't change with our reactions what we produce or their energy. We're still going to go to Y. I'm sorry, we're still going to go to X, but we will lower the activation energy enough that we can make this happen. Now, this is endergonic, so energy has to be input into the system to make this process go. So sometimes this energy comes from light, sometimes it comes from ATP. There are a number of ways that we can supply the energy needed to make substrates that have greater energy. And we know this because our synthesis reactions, which we know happen often when we're synthesizing molecules, because we're creating new bonds, it means that our molecules have more energy in, dang it, product. Hello, sorry about that. So when our products have more energy than our reactants, we have to input energy into the system to create these new bonds to store this additional energy. And so, but we still need enzymes to help perturb the bonds so that the new forms, the new products can be made. So let's talk a little bit about how we control proteins, and because most of the enzymes we'll be looking at this semester are proteins, how we control enzymes. 3 ways that we will see over the course of this semester of how we control proteins, how we control enzymes, and by control we mean regulate, whether they're on or off. Feedback inhibition or feedback stimulation, depending on the situation. We can also control proteins and enzymes by their phosphorylation state, whether they have a phosphate group attached or not. And we have some proteins and enzymes that are controlled by binding ATP or ADP, or in other cases GTP or GDP. So we'll see these examples over the course of the rest of the semester. In the case of negative feedback inhibition, so let's consider we have a reaction pathway here where substrate A becomes product B, and then that becomes substrate for the next reaction. We produce product C. We may have a different enzyme that is going to convert B into product X, and then X becomes the reactant for the next reaction, and we have a reaction to convert X to Y, and then Y to Z. Each of these are going to have different enzymes controlling the reactions because, like we said, the enzymes bind the substrate, and because that binding is very specific, if you have a different reaction, then you're going to need a different enzyme to bind and convert to the different product. So, if we have a synthesis pathway where A produces B, produces C, B produces X, then Y, then Z, if we build up enough Z so that we don't want any more produced, Z is going to find a way to impact the enzyme, the first enzyme that is unique to its production line. So Z is going to turn off the enzyme that converts B to X so that we don't get any more Z. It would not make sense for Z to feedback and turn off the enzyme that produces this reaction here, A to B, because then that would affect the assembly of C, and we may not have enough of that yet. So for negative feedback inhibition, when you've got these complex metabolic pathways, their regulation works where the end product of a line, once it has built up and we have enough of it accumulated for whatever the job is, it will do a negative feedback inhibition on the first enzyme unique to its synthesis pathway. And that's important to understand why it would impact there and not here. If Z impacted and turned off the enzyme that does X to Y, then we would still get a buildup of X, and we don't need X, right? So we still need B, so we don't turn this one off, but we do turn the first one that's unique to its pathway. Okay. Here's an example where the final product of a pathway for this enzyme is a molecule called CTP, cytosine triphosphate. And it is going to go back and inhibit the first enzyme unique to its pathway. If this is our enzyme here and our active site is shown here, and so it's on, what this example is showing an allosteric inhibition. Allosteric, A-L-L-O-S-T-E-R-I-C. Allosteric means away from the active site. So it's an indirect inhibition by the product binding to the enzyme away from the active site. It still changes the conformational shape of the active site in such a way that it can't bind substrate anymore. So this turns the enzyme off when our final product binds it. So this is the typical way, this is a common way that feedback inhibition can happen. You can also have direct binding of the product to the active site, which is a direct inhibition, but there's also this indirect kind. Either way, when the enzyme can't bind substrate, it's off. So here is a complicated little chart. We have aspartate being converted by a variety of enzymes into aspartyl phosphate. We have an enzyme that converts the aspartyl phosphate into aspartate semialdehyde. From the aspartate semialdehyde, we can convert it into homoserine. We can also run a line out to produce lysine, which you should recognize as an amino acid. And of course, the aspartic acid, serine, and related homoserine. Homoserine can be converted into threonine and isoleucine in this way, methionine in this way. And so if you think about how these loops work, the pink lines are showing you the negative inhibition. So for example, isoleucine here feeds back on the first enzyme that's unique to its production pathway, shutting off its own synthesis. Methionine, similarly, feeds back on the first enzyme unique to its production pathway. Is, for example, here for isoleucine, it inhibits this enzyme here unique to its pathway. It does not go up and inhibit this one because that would knock off threonine as well. So it's complicated, but that's the essence of negative feedback inhibition. Another way that we control proteins is with their phosphorylation state. So here's our protein in green here. One of the amino acids is highlighted here. It's a serine. And you see the R group of serine here in pink. Here is that same protein, and here is its serine with a phosphate group attached. So we burn an ATP molecule, use that third phosphate to attach to the serine residue, pull that hydrogen off, and attach our phosphate here. We're left with ADP, adenosine diphosphate, and that third phosphate group was used to attach to the protein. So now we say that this protein is phosphorylated, and the enzyme that phosphorylates this protein is called a protein kinase. Kinase is a term for an enzyme. ASE is our hint that it's an enzyme, and kinases are proteins that phosphorylate, or enzymes that phosphorylate molecules, typically proteins. So this is a serine kinase because the kinase attaches the phosphate group to serine residues on the protein. To get that phosphate group off, we need a different enzyme called a protein phosphatase, and it is going to pull that phosphate group off and put a hydrogen back on, and we've got now back to our unphosphorylated protein. So for some proteins, when they're not phosphorylated, they're off. When they are phosphorylated, they're on. For other proteins, they are on when they're not phosphorylated and off when they are. And so there's no strict pattern to predict on this example, but the business end of what you want to remember here is that kinases add phosphate groups by burning an ATP molecule to get that phosphate, and phosphatases remove the inorganic phosphate here. So kinases add phosphate groups, phosphatases remove them. Either way, we're controlling the on-off status of these proteins. So this is just showing that kinases add phosphates, phosphatases remove the phosphates. For some proteins, being phosphorylated turns them on. For other proteins, being phosphorylated turns them off. and it just depends on the protein. Finally, GTP binding. For some proteins, they have a binding domain for GTP. GTP is guanosine triphosphate. Now, unlike phosphorylation with proteins, where for some proteins, when they're phosphorylated, they're on, and for others, they're off, when a molecule that has a GTP or GDP binding site, when it is bound to GTP, it is on every time. This is a rule, there are no exceptions. So for GTP binding proteins, when GTP is in the binding site, that protein is on. To get GDP in the binding site to turn it off, you simply get hydrolysis of the GTP. That third phosphate falls off and you're left with guanosine diphosphate, and now this protein is off. So whatever it did while it was on, now that function is shut off. Very slowly, the GDP will pop off and will be in an inactive state. And to get this protein reactivated, we have to put a fresh, complete GTP molecule on. And now it's on again. It won't stay active very long because the guanosine triphosphate is fairly unstable. The more stable form is to let go of that third phosphate group and hydrolyze down to GDP, and then we'll be in the off position. Typically where we're going to see GTP, GDP binding is the same drill for ATP or ADP binding. We're in signaling pathways, so that once we turn it's like hitting the dominoes. This is the first domino. Once we hit the dominoes, then the rest of the chain of dominoes takes over. So we don't need this turned on for very long, just enough to get the cascade started, and then we can turn it right off. So what's different here, and completely distinct from kinases adding phosphates and phosphatases removing them, here we put a fresh GTP molecule on. It's on very briefly. It quickly hydrolyzes down to GDP and it's off. To get this protein reactivated, we have to completely pop off that GDP so that it's now completely missing from this binding site, and we have to put a completely fresh GTP molecule on to get this protein reactivated. Okay? We are not, this is what we do not do. We do not phosphorylate the GDP to make it GTP. That's not how it works. We have to completely get rid of that whole GDP molecule and put a completely fresh GTP molecule on there. It's the only way we get this to work. Same drill for ATP, ADP binding to control proteins. So we've talked about 3 ways that we control proteins: feedback inhibition, their phosphorylation state with the kinases and the phosphatases, and then GDP, GTP binding. Ta-da!